All right. Good morning, everyone. I am Kailing, the Regional Field Application Scientist of Azure Systems. Today, we're in the Gene Expression Analysis webinar, which we will be focusing on relative quantification strategies. Before we start, let me introduce Azure Systems to you. We are a team of highly experienced scientists, engineers, and entrepreneurs dedicated to accelerating your science. Our company founded in United States, California, California. Our mission is to giving you raw data that you can rely on using simple workflows that you can trust. Azure System is founded by Arnaud Shiji. He's our CEO and chairman. He's also a venture investor on innovative companies. And Dr. Diping Che. Dr. Dimitri is our Executive Vice President of R&D. If you look into his past experience, he has a lot of experience in developing protein imaging system in the Illumina and Alpha Innotech. Azure System is a fairly new company. The company founded in 2013. All the core members are coming from Alpha Innotech. We have a few instruments in our company portfolio, for example, Sapphire Biomolecular Imager, mainly for laser scanning system, Azure Imaging System for Western blotting imaging, and also Cielo Real-Time PCR system in the genomic market. Right. So today agenda, we will be focusing on relative quantification strategies in qPCR analysis. I'll be talking about 3 specific strategies for you. The first one is relative quantification with standard curve, comparative CT, or what we call as delta CT, efficiency corrected calculation model, or we call as PAF models. In this webinar, I'll be talking about their principles, basic calculation, and also some publication example on how people improving these calculation. First, let us look at the quantification strategies in real-time PCR. In general, there are 2 strategies that can be used in analyzing qPCR data. The first one is rather straightforward, absolute quantification, where we relate the qPCR signal, for example, CQ value or CT value, to a certain input copy number using what we call as calibration curve or standard curve. Second is relative quantification. Relative quantification, this is where we have more than one group or more than one gene involved. We're going to measure the relative changes in gene expression level. This is a simplified diagram from published by the Paul, okay, Michael W. Paul back in 2004, where it talks about quantification strategies in real-time qPCR. So here it's divided into 2 major quantification, absolute quantification and relative quantification. So here I said absolute quantification, you normally either need to have external calibration curve or we call it standard curve for one color or multiplexing assay. And this quantification will be reliable if and only both unknown sample and standards have the same amplification efficiency. In the previous webinar, we have been focusing on this absolute quantification, where we mentioned that the choice of your the choice of the calibrator or the template for generating the standard curve is very, very important, because that would tells you and whether how tells you the exact CQ or the DNA copy input for that you count back to your target gene. Right. So in this webinar, we will be talking about relative quantification. Okay. In relative quantification, we have 2 major category. First one is meant for normalization. This will involve with or without with reference genes mostly, and another one is relative quantification with standard curve. Right, so let us go through the first one, relative quantification with standard curve. This method, also called as E method, can produce more accurate relative quantification data as it is based on the true PCR efficiency for both your target gene and housekeeping gene. So in general, this is much more easier than absolute standard curve method because the quantity is expressed relatively to the standard. Rather than you have an absolute amount, you are going to input it as a relative amount, whether in dilution or arbitrary number. But it can be either an untreated control or time point zero, let's say, or a healthy individual. Nucleic acid template containing target genes and housekeeping gene can be used to prepare standards by serial dilution. These standards must be accurately diluted as their relative dilutions are important to plot the relative standard curve and quantitate the relative expression with the unknown sample. So both unknown sample and the standards must have the same dilution, so they will be calculated accurately. Relative standard curve uses a set of relative standards with unknown samples. So you can use unknown samples to generate your standard curve. The standard will be 100% or one time, where the other unknown samples are expressed as n-fold differences related to the standard. qPCR users need to prepare standard curves for both target gene and housekeeping gene in order to do normalization. This is a simplified diagram showing you how what's the concept of relative quantification with standard curve. Basically, you have a standard. This standard can be either from your pooled sample from all your experimental sample, including your control, or anything that is relevant, like say healthy individual, time point zero, or even like non-treated control. That can be your standard. You use this standard to produce, here will be two sets of standard curve, one for target gene, another one for your housekeeping gene. Okay, now you have two standard curves generated from a serially diluted standard, which has the same dilution factor as your unknown sample when you do your qPCR assay. With this, when you run your qPCR assay with in the unknown sample at the same dilution factor, you will get the CQ value for both target gene and also housekeeping gene. From this CQ value, you use it to plot get the arbitrary amount or copy number in the standard curve. Okay, the equation or the calculation step by here. So normalized target gene in 1 in 20 diluted standard will be A, right? So this is normalized copy number, target gene divided by housekeeping gene, okay? And you have another one, normalized target gene in 1 in 20, okay, diluted unknown sample, which is B, which is X, the amount of X calculated based on X CQ value, and the number calculated based on Y CQ value. That's you get your ratio. Well, how to get your fold differences, or say the relative expression, is you divide the unknown sample, the ratio, divided by the standard. That's it. So this is how you get the fold differences in comparison to your standard. This is how it looks like in Cielo Manager, a software, analysis software, and also managing software that can control Azure Cielo Real-Time PCR systems. Where you can see we have a few samples over here, sample 1 and sample 2, and also their standard, or say standard wells. Each for gene CMYC and GAPDH, where GAPDH is your reference gene and CMYC is your target gene. With that, you will generate a standard curve. Then the CQ value from the unknown sample, here you can see it, okay, will be calculating the starting quantity at this standard curve, where you get your quantity all calculated here. Then the data will be recalculate the ratio, okay? So you will get this image over here, the graph. So here you will see the target of CMYC in sample 1 and sample 2, okay? And then the normalized quantity mean, and here will be relative quantification, here expressed in 4. Here will be 1, okay? This is 0.688, lesser than the sampled one. There's a lot of publication out there that is trying to improve this calculation method as well. There are also some publication who addresses certain crucial issues when you use relative quantification with standard curve. For example, this publication in this year, published in Clinical Chemistry, where it says that efficiency correction is also very important and is required for accurately quantitate the qPCR analysis, even though we use a standard curve. So we need to have both in dilution and actual dilution. So if you're interested, you can always check this paper out and see how they can improve such calculation method. Right, next, let us move to a very famous method, which is comparative CT method. This method, also called as delta CT or delta CT method, without any efficiency correction. These are the equation over here, how we get the data. So here, work out delta CT, okay? Delta over here will be the changes between the target gene minus the reference gene, okay? And the delta CT will be the changes of the changes itself. This method directly uses the threshold cycle, either we call it CT value, or quantification cycle value, or CQ value. The difference over here is CT value is the value that you calculated from the software based on threshold setting. So CQ value will be calculated based on the software, either regression mode that calculate the CQ value for you. They are basically the same, which is the data you get from qPCR qPCR run. Comparative CT method or delta CT method, it assumes 100% PCR amplification efficiencies across all samples. This applies to all your sample, including your control, including your negative control, including your positive control as well. In reality, it may not happen in that way. So one very key important, I'll say important thing that we need to be aware is amplification kinetics of the target gene and reference genes, assay, must equal. This when you, let's say, between target gene, we were talking about primal efficiency and also qPCR assay efficiency over here. When you have different efficiencies between, let's say, assay and even the primer itself, this will generate error in this calculation method. The reason being is explained by this graph. This graph will tell you, let's say we have a control sample indicated in the dotted lines. The expression here will say and GAPDH and the TNF alpha over here. Okay, the magnitude is different than what you can see in the treated group. Okay, the CQ values are different. Therefore, we need reference gene to do the normalization, and then these two, okay, GAPDH and TNF alpha, their primal efficiency must be similar in both control and treated samples as well. In that, if you do comparative CT method or delta CT calculation, you must do validation assay for both target and reference gene with serial dilutions to show both of these primer sets have the same efficiency, and look at how the delta CT varies with the template dilution. How you do that is you do your normal, you have your standard, same standard, then you produce, you go through your qPCR run, you got your CQ value, then you plot the result with x-axis is the CT value over here, and the y-axis will be, sorry, this is inverted. So CT will be at the y-axis over here, differences in the CT, target minus reference for each dilution, and the y-axis will be the log input concentration for each of the dilution of the template, either cDNA, RNA, and so on, depending on your application. Right, so this graph is what we call as validations of the two delta method. Okay, this is to show that your delta CT is true when the dilution increases, because we know that when you have primer sets and even the qPCR run, when the template get diluted, if your primer is not 100%, having 100% efficiency, the efficiency may drop in certain dilution. So it's important for you to validate this when you do a comparative CT calculation method for your qPCR data analysis. How we see is if the absolute value of the slope, we're looking at the slope over here. The slope, if this is lesser than 0.1 and close to 0, like this graph over here, this graph is 0.04, which is lesser than 0.1. The efficiencies between the target and the reference genes are similar. So you can use this comparative CT or delta CT calculation for your qPCR data. Right, say in the figure, the slope is 0.0471. Therefore, the assumption hold. That means both target genes and also reference gene having the same amplification efficiencies or similar. Therefore, you can use the calculation method. Let's say once you plot this graph and you realize that the slope is more than 0.1. Let's say you have 0.15 or 0.2. The assumption will not be hold anymore. That means both your target genes and reference genes are not having the similar efficiencies. With that, you either need to do your data analysis via standard curve, means relative quantification with standard curve, okay, or absolute quantification with standard curve. Alternatively, you can improve the qPCR efficiencies by designing new primers or even optimize your whole qPCR assays to achieve similar efficiency between the target and reference genes. So this is a validation assay, quite important if you want to do delta CT value calculation method. Right, so this is the analysis example of how it should analyze. This is published by Leva in 2001. This is a very famous paper that a lot of people referring to up until today, which addresses delta CT, what is the basic, what does it concept, and the complete concept behind it. So this is where you look at, you have multiplex assay, both target and reference genes are amplified in the same well. So this is how we should calculate it. You have your replicates over here, okay? CMYC versus GAPDH. These are multiplex assay within the same well, okay? You get the CT values, okay? Then how you do that is you calculate the delta CT, and means the average CT, okay? You divide this gap CMYC divided minus the GAPDH CT, where you get all these value, then you mean average them, okay? I'll also get the standard deviation. Then you get 6.93 plus minus 0.16. Same goes with your kidney. So kidney in this sense will be your control, okay? Oh, sorry, brain will be your control. Kidney will be your, let's say, treated or tested sample. Right, so now you get your delta CT, which is the differences of your target genes minus the reference genes. then you need to calculate your delta CT. Delta CT means the average of DCT minus the average of the CT brain. Brain is the control here, so you will get 0. This will act as your control, will be shown as 1 or 100% in your data later, in the graphical graphic or bar chart later. How you calculate it is you 4.47 minus 6.93. Now you get minus 2.24. Right. So how you get the normalized amount relative, there's a relative quantification, okay, to the brain, which is a control sample, is you do 2 to the power of minus, okay, delta CT. So 2 to the minus 2 to the power of minus 2.7, you get 5.5 volt. Okay, this is the relative quantification here. This applies when you have multiplex assay. If you have different assay, let's say you have you done the entire example in the same plate, but target genes and the reference genes are amplified in different well, the calculation will be different. Do bear in mind this. Okay, if you're interested to know, the publication will tell you how exactly you can calculate it based on different qPCR assay that you did for your experiment. Next one is a much more detailed display in Excel sheet over here. Okay, as a demonstration of two delta CT calculation method. Okay, to compute the relative gene expression from qPCR data. In this data, we have a different set. You have different concentration of treatment. Here's a silencing RNA treatment over here compared to the NTC. Okay, so NTC will be your baseline, will be your control. Same thing, you minus, you get the delta CT or delta CQ over here for each of these replicates. These are corresponding to each replicates. Okay, then you get your average CQ expression over here, right? And the standard deviation calculated. So you get calculation mean. This is a for each of them. Then how to get the delta CQ expression is you here mentioned here is G1 okay, divided by G10. So the mean delta CQ of your treated sample divided by the delta CQ of your control. That's how you get your delta CQ expression, right? And then the standard deviation, I will not explain more here. Okay, so this is how you basically calculate it. And since this is a silencing assay, okay, a knockdown assay, you need to calculate the knockdown percentage. How to do the knockdown percentage will be 1 minus the delta CQ. That will achieve 97% of knockdown in this case. Let me show you how it looks like in the Cielo Manager software, where this is what we call as we are not doing multiplexing here. This is single plex, okay? 3 wells for the CMYC, another 3 wells for the GAPDH. You have wild type as your control, okay? So when you plot the data will be shown like this. You have wild type as serving as your 1, which is also we can call it 100%. Then you have the fold change or related quantification here. So you can see the target, which is the step one target in the wild type sample, serve as 1. And then the step 1 in the ChemK2A samples will have 107 fold changes differences as compared to wild type control. So this is how you interpret the data. Most of the software nowadays can auto compute the result for you. So you can also, besides this, you can also rely on Excel sheet to calculate it for you. Right. Even though this delta CT calculation method is quite established, I'll say, and a lot of people are actually using it because it's easy. You only need to have the CQ value or CT value, so it's rather easier to use. But definitely we have some improvement on this calculation method. So this is a paper published in 2013, okay, and where they address that, we have account for qPCR efficiency, we have in qPCR assay, there's a lot of factors will influence the data that you get from the qPCR assay. So with that, they do mention that it's very important to correct with the efficiency. Right, this is the efficiency correction on top on the delta CT method. The calculation is a bit lengthy over here. I will not go into details, but if you're interested in how to improve the delta CT calculation, you can always check this publication. Right, let's move to the third quantification strategies, which is efficiency corrected calculation model, or we call it as PERF model. This model combines both gene quantification and normalization into a single calculation or single equation. And it uses amplification efficiencies of both target and reference genes to correct for the differences between the two assays. Here is some of the, I'll say the equation over here mentioned for this. So since this is a calculation method corrected with the qPCR efficiency for both target and reference genes, we have the calculation will be different based on one sample. If you calculate based on one sample, that will be easier. That means your efficiency, I'll say here will be efficiency times the delta CT target control minus a sample. So this is a bit mouthful. Later I'll show you in the explanation. Okay, so this is the equation it looks like when you have one sample. So this is based on multiple samples where you will get mean for control and samples. And this is also based on multiple samples and on multiple reference gene as well. This is where you get the reference index. The reference index will be generated by at least 3 reference genes. Okay, so this calculation method is a bit more complex in that sense, but it suits for most of the application where you have one sample, more samples, okay, and multiple samples and multiple reference genes. So you do see that in this quantification strategy, we do need to use reference genes to do normalizations, right? Where the target gene expression is normalized by one or more reference gene expression. So that's why reference genes are very important. One very crucial problem of efficiency corrected calculation method or PERF method is we need to choose the correct reference genes. Because as we know that reference genes are available that we can allow us to choose, sometimes depending on your experiment or your application, the reference genes expression, they do varies. They vary significantly and they influenced by either your treatment, tissue type, or even individual differences. Some of these may have been addressed by publication or certain researchers. They address this issue, they find this, they have to look for new, let's say a newer or a better or more stable reference genes. Some are yet to be verified because maybe you are doing a certain, let's say you're using a certain treatment that nobody has used before. So you will be unsure whether your reference genes is really stable. Therefore, for this efficiency corrected calculation model, you need to choose the right reference gene to normalize with. And you need to choose the best reference genes for gene quantification assays. I do want to do at least the multiple reference genes normalization. You can do that as well. There will be at least 3 reference genes, one for the highest, very, very high expression, one for middle range expression, one for lowly expressed gene. Okay, you can do that. So you have multi-corrected calculation over here. Best is to validate, all in all, okay, best is to validate the stable expression of reference genes with the serial amplification efficiencies with a serially diluted template. This is very important. And also do count in the factors of real-time and also PCR inhibitors in the entire assay. Because the qPCR itself is not only you're talking about primers, we're talking about templates and everything. The qPCR is also important. Okay, if you have a more, say, complex, let's say one-step qPCR assay, is much more complex. So you also need to take these into account as well. This is in order to fit the calculation method into the assumption of target and the sample, both target and sample, amplify at the similar efficiencies. So qPCR, you need to make sure all of your runs and all of your assays are at the similar efficiencies. Try to either optimize it to design your primer or change a certain, I'll say, change certain parameters to or reagents to make them having the same similar efficiency across the entire qPCR assay, different plate, different samples, control and treated, and so on and so forth, and also different set of targets, target genes and reference genes. All right. So here's how the calculation works. Basically, you would need to generate a standard curve for each of these target. You will generate the standard curve for your reference genes, okay, and also your gene of interest. From there, okay, using a serially dilated template, from there, you calculate the primer efficiency. Here's a primer efficiency in percentage, okay? Here's a primer efficiency based on the slope itself, okay? Here, if you can see the efficiency for primer, the best will be at 2. 2, at the value of 2 means you have 100% efficiency. Every single cycle, you will definitely get 2-fold increase as the cycle goes, right? But in reality, it doesn't work that way. So for efficiency corrected calculation model, you need to generate standard curve for each of these primer set and then get the efficiencies calculated for each of these primer set as well. Right. So then you have your target gene and gene of interest at your sample, control sample, and treated samples. With that, you get the mean CQ. This is for multiple samples over here because you have 2 controls and 2 treatment, right? So you're using mean CQ over here. Okay. Then how you get the relative quantification as compared to, okay, within the sample itself is this. Means the calculation goes like this. Efficiency to the at the power of, okay, bracket reference genes in the controlled sample minus the reference genes in the treated samples. So this is how it looks like. Okay, so you have efficiencies 1.94 to the power of bracket 2.65 minus 25.6. Therefore, you got the quantification fold changes, or say, over here in 0.53. Right, so this is how the calculation goes. We call it fold difference value results. Right, so therefore we have 3 relative quantification strategies over here with with standard curve. Second is a delta CT method. Third is this efficiency corrector calculation method or model with PALF models. You have to bring your attention to this publication, okay, published by Luca Boha, that's comparing this method for relative quantification of gene expression using real-time PCR. In this publication, this researcher comparing these 3 relative quantification strategies, okay, standard curve, with standard curve, delta CT, and efficiency corrected. So there, in their paper, there's a lot of data. So I'll show you some representative data over here. Right. First thing, definitely they are generating a standard curve for qPCR efficiencies calculation for both target genes and also reference genes in order to get the primer efficiency calculated over here. And just to show that the primer's efficiencies are fine. Right. Second, this is much more relatable to delta CT method. This is a validation curve. for delta delta CT method, okay, where they plot the y-axis with the absolute delta CQ value and plotted against a log copy number at at x-axis over here. As you can see, okay, right, you have a standard curve was created each time a total DNA quantification was performed, so you get the accurate calculation. And here you have the slope of this standard curve, okay, for the standard curve and efficiency calibrated method to get here primal efficiency and linear dynamic range. And this, you will get the slope, which is supposed to be 0.1, okay? So this is closer to 0, lesser than 0.1. That means the efficiency for both targets and reference genes are at the similar efficiencies, okay? Right, slope of these two indicating the slope of this indicating that the assumptions of equal target genes and the reference genes efficiency was correct. All right, so you need to make sure this is established if you're using delta delta CT calculation method. Hence, the design primers, the two set primers, reference and the target, okay, are both suitable for related quantification using delta delta CT method. Okay, so this is much more related to delta delta CT. Right, we have calculation for all of these methods. They are basically the same set of sample, but just different calculation. You have efficiency calibrated method and the delta delta CT method over here. Right, I'll not go into details on how to calculate this, okay, because it's quite hard without the mathematical equation. So, but I will bring your attention to this, I'll say the final result, where they compare the analysis, the three different analyses with a same set of data. Right. So you have the standard curve method indicated in this dark color bar over here. This is the fault changes calculated. And then these are fault changes calculated by efficiency calibrated method and also the delta delta CT method over here. Right. So we have the standard curve method, efficiency calibrated method, and the delta delta CT method. They all of them generate comparable and reliable results when all necessary conditions for each methods are fulfilled. Like I say, when you use delta delta CQ, you need to have the validation curve to show that both the target and reference genes having the same efficiencies. Okay, and same goes for the standard curve. You need to have the good. You need to choose the standard to plot the standard curve and also the efficiency calibration. That one is easier because you just need to generate standard curve using the primer set and the template. There you get the primer efficiency calculator. Right. So as you can see that these three, they are not having any using these three relative quantification strategies. The differences of expressions calculated, okay, are not very much different when you have high expression changes as compared to control. But when you have low expression changes or differences over here, these three methods will start to show some pros and cons. Okay, in general, okay, when you quantify small differences in gene expression, for example, this in sample 3 and sample 4, where you have slightly lesser expression or slightly more higher expression, okay, the delta delta CQ method should be avoided. Okay, both standard curve and efficiency calibrated model, okay, or method are more suitable, okay, to determine very small differences in relative gene quantification, right? Where the most accurate method will be standard curve method, right? So the arrangement goes like this. The best one is standard curve method. Second is the correct efficiency corrected model. And the final one will be delta delta CT method. All right. When you try to quantitate small differences between the samples. Okay. All right. Okay. So here pretty much a very simple presentation about the relative quantification strategies in qPCR analysis, where we talk about three major strategies over here. The first thing is standard curve. Second is a delta delta CT model. of calculation, and the final one is efficiency corrected calculation models, where we talk about the principles and show you certain calculation, and the publication showing you which one you should pick and what you should do with them. Right. In this, let me introduce Azure Biosystems, Azure Cielo Real-Time PCR systems to you. Okay, in related to this webinar, this is an open platform supporting both dye and prop-based chemistry in qPCR reactions. In terms of performance, it has very unique optical design to deliver more sensitivity and reproducible data. Okay, and this is also flexible due to the nature of an open system or open platform that's engineered for a wide variety of qPCR application. It also proven to deliver trustworthy data, okay, in the sense of reproducibility and uniformity. You can run a qPCR protocol by using Cielo, this machine, as a standalone machine. It has very simple touchscreen assay setup, and no external PC is required to operate it. But data analysis will require PC, which is a Cielo manager, to analyze the data. With that, you can also run or control this system from a PC that is connected via either Ethernet, LAN, Wi-Fi, or USB. These Azure Cielo Real-Time PCR systems, it contains or designed around individual well scanning. So it scanned individual well, where you have two sets of 16 fiber optics, okay? One set coming from the high-powered channel-specific LED to excite the fluorophores in the reaction, qPCR reaction, and another set to deliver the signals to the detector camera, right? And these 2x8 fiber optics or 2x8 scanning over here to ensure that you have all the specific wells on the across the entire qPCR plate are evenly illuminated. So even illumination, and then you do not have excessive light shine outside the well. Therefore, the background signals will be minimized, and you won't masking the fluorescent signals coming out from the reaction itself. This is pretty much different than a conventional PCR system that use one single light source shining on the entire 96 well plate, and then also one detector camera to capture the signal in a one single image. So this is different than that, right? And one key note over here is these optic fiber designs, okay, avoid the need for passive reference lines. You do not need passive reference lines such as rocks to normalize or calibrate the optical path variation when that happen in the conventional PCR system. Azure Cielo Real-Time PCR systems comes with two configurations. We have Cielo 3 and Cielo 6. 3 stands for you can do up to 3 multiplex in a single reaction or single well. In Cielo 6, you can do up to 6 plex reaction in a single well. And these are the list of the dyes or probes are already calibrated in the machine. So you do not need to have additional calibration using the machine itself. But if you have certain custom dyes that is fit into the excitation and emission wavelengths over here, you can always calibrate the custom dye into the system by using just a one step, prepare the plate and insert it. That's it. It's very simple. Right. So if you look at the filter wavelength for both excitation and emission wavelength, you will see that all these channels has been distinctly differentiated one and another to ensure there will be low to none crosstalk when you do multiplex reaction. Azure Cielo Real-Time PCR machines, it has been consistently proven and also validated for interwell uniformity and across the entire 96 well plate. So the sample you put at A1 well will not have different data than the sample you put it, let's say H10 well. The same interval uniformity detection also tested across different machines to ensure that there will not be any instrument-to-instrument differences. In a qPCR reaction or PCR reaction, temperature also very important, where In this machine, we have 6 Marlow-Peltier blocks to ensure entire uniformity and accurate temperature to run the qPCR reaction. Ability to detect twofold differences in gene expression and multiplexing without and low signal crosstalk, melting curve analysis for you to look at whether this amplicon is specific or not at 3 different resolutions. The resolution is up to 0.1, 0.1, 0.5, and 1 degree Celsius per second. Reproducibility from run to run. The machine has been tested for continuous 1,000 runs. Okay, then we have plotted every 100 runs to show that the CQ and the coefficient of variance are very, very low. And the broad application compatibility, this is from the calibrated die list itself. So this is an open system. We do not have any proprietary dies that you have to couple with this machine. Right. Here's some of our selected customers across the entire world. We have, let's say, PerkinElmer, SynLab, Dentellab, Only Lab. We have research institute and university as well. We have University of Master University, University of Sherbrooke, China Medical University in Taiwan, okay, and University Illinois, Chicago. Coming from Singapore, Sari Roti from Indonesia as well. Here's some customer reviews on Select Science, where they review how this machine helped them in terms of solving difficult situation. For example, from University of Sherbrooke here, this group researcher is having some difficulties in amplifying difficult target in a consistently low DNA input amount, which they cannot max out the input amount anymore. So their Cielo is able to consistently target their amplify their difficult targets at a low input DNA amount as well with an earlier CQ value. Right. So here's this brochure that is simplified and show you what is in these Azure Cielo Real-Time PCR systems, the operation, the durability, thermal performance itself, optic connectivity, chemistry, speed, capacity, and also sensitivity. We are also 3 application notes available on the website, or you can request from Unimed, where we address why this machine and how it achieves multiplexing, okay, without or low no crosstalk, and single copy amplicon detection over here, where we push the limit of the machine, and also why this optical design do not need for passive reference die. Here's some in the brochure. I will highly recommend you to read on it. It would actually tells you what is happening in a qPCR machine and what technology has been evolved for so far. Okay, thank you everyone. I'll open the floor for Q&A section before we move on to a Kahoot and also a Kahoot session, game quiz session. I'll pass the floor back to Priscilla.
好，谢谢凯宁很精彩清楚的介绍。那不知道大家对于今天的讲座内容有没有什么问题呢？都可以直接在这边提出，用中文或英文都可以。那或者是你也可以写在旁边，右手边有一个 chat box， 对，你也可以把您的问题写在上面。对，那我们稍等一下，让大家想一想有没有对于今天的内容有没有一些想要提问的部分。
If you have any question, please type in the chat box. Okay, so I can address your question in this.
中文或英文都可以哦。
Yes, if I Mandarin is not able to understand your question, Priscilla will help to translate as well. All right, I think we are almost reaching the time. We do not have any question. I don't see any question from the chat. Let me put up the Q&A and oh no, okay. So I'll assume that there's no question from the floor. Priscilla, shall we move on to Kahoot session?
对，但稍等一下。
OK
这边大家可以看到那个 chat box 上面，我有提供一个 feedback 的问卷连结。那麻烦大家可能帮我花个1分钟的时间，填一下您对于今天的线上讲座的一些想法，或者是之后你们有这个讲座回馈表，或是之后你们有没有什么关于QPCR 想要听的主题，也可以留言告诉我们。对，那我们会针对大家有兴趣、感兴趣的主题，然后再规划之后的线上内容。谢谢。好，那